Chromosomes. Chromosomes are long, thin structures found in the nucleus of cells, appearing like intertwined threads under a microscope. Each chromosome consists of tightly coiled DNA wrapped around proteins called histones, which help maintain its shape and regulate gene activity. Chromosomes contain genes, specific sequences of DNA that code for traits like eye color, height, and metabolism. Genetic information in chromosomes is passed from parents to offspring, determining inherited traits. Chromosomes store instructions for cell growth, division, and function, ensuring proper development and maintenance of life. In 1882, German biologist Walter Flemming first observed and described chromosomes while studying cell division under a microscope. He used dyes to stain cells, making the thread-like chromosomes visible during mitosis. His work laid the groundwork for understanding how chromosomes carry genetic information and pass it from one generation to the next. The word chromosome was first used by German anatomist Heinrich Wilhelm Waldeyer-Hartz in 1888. Chromosome comes from the Greek words chroma and soma, meaning color and body. This is because chromosomes absorb certain dyes, making them visible under a microscope. Waldeyer's naming helped standardize terminology in cytology, aiding future research in genetics and cell biology. Characteristics of chromosomes. Numerical variation. Different species have different numbers of chromosomes. For example, humans have 46 chromosomes, while fruit flies have 8 and rice plants have 24. However, the number of chromosomes does not necessarily determine the complexity of an organism. For example, some ferns have over 1,000 chromosomes, while humans have only 46. Dynamic structure. In the interphase, chromosomes exist as a loosely coiled chromatin network, allowing gene expression and DNA replication. As the cell prepares for division, the chromatin condenses into visible, rod-shaped chromosomes, ensuring an organized and even distribution of genetic material. Size variability. Chromosome size can differ within a species. For example, some human chromosomes are longer than others, and between species, with some organisms having much larger or smaller chromosomes. Larger chromosomes generally contain more genetic material, but their size is not always directly proportional to gene count. In mammals like great apes and paramyscus rodents, smaller chromosomes often exhibit higher recombination rates and greater nucleotide diversity, leading to increased sequence divergence compared to larger chromosomes. DNA packaging. Histone proteins play a crucial role in packaging DNA into compact units called chromatin, which not only prevents DNA tangling and damage, but also facilitates gene regulation. Modifications to histones can alter chromatin structure, thereby controlling gene expression by either making genes more accessible to transcription machinery, turning them on, or less accessible, turning them off. Self-duplication. Before a cell divides, chromosomes replicate to ensure each daughter cell receives an exact copy of genetic information. This replication occurs during the S phase of the cell cycle, where each chromosome forms an identical sister chromatid joined at the centromere. The accurate duplication and separation of chromosomes ensure genetic continuity, preventing mutations and maintaining species integrity. Functional regions of chromosomes. Chromosomes may consist of several important regions, such as the centromere. This is a specialized region of the chromosome that plays a crucial role in cell division. It serves as the attachment site for spindle fibers during mitosis and meiosis, ensuring that chromosomes are properly segregated into daughter cells. Without a functioning centromere, chromosomes may not be evenly distributed, leading to genetic disorders. Telomeres. Telomeres are the protective caps found at the ends of chromosomes. Made up of repetitive DNA sequences and associated proteins, they prevent chromosome ends from deteriorating or fusing with neighboring chromosomes. Each time a cell divides, telomeres shorten, which is linked to aging and cellular lifespan. The enzyme telomerase helps maintain telomere length in certain cells, such as stem cells and cancer cells, allowing them to divide indefinitely. Heterochromatin. These are tightly packed forms of DNA that are generally inactive in gene expression. It is found in regions like the centromeres and telomeres, where its condensed structure provides stability and prevents unnecessary recombination. There are two types of heterochromatin: constitutive, which remains permanently inactive, and facultative, which can switch between active and inactive states, depending on the cell's needs. Euchromatin. These are loosely packed active regions of the chromosome where most gene expression occurs. Unlike heterochromatin, euchromatin is accessible to transcription factors and RNA polymerase, allowing for the production of messenger RNA. This form of chromatin is essential for normal cell function, as it enables the expression of genes necessary for growth, metabolism, and differentiation. Types of chromosomes. Chromosomes can be classified based on their function in an organism's genetic makeup. Some chromosomes determine the biological sex of an organism. These are called sex chromosomes. In humans, there are two types, X and Y chromosomes. Males have one X and one Y, while females have two X chromosomes. The Y chromosome carries genes responsible for male traits, while the X chromosome contains essential genes for both sexes. In birds, the system is reversed compared to mammals. Females are heterogametic, having the ZW chromosomes, and males are homogametic, with the ZZ chromosomes. All the other chromosomes that are not involved in sex determination are called autosomes. Autosomes contain genes that influence physical traits and biological functions. They carry the majority of an organism's genetic information and play a crucial role in the overall development and function. Humans have 22 pairs of autosomes. Unlike sex chromosomes, autosomes are present in identical male and female pairs. Based on structure, chromosomes can be classified based on the number of centromeres they possess. Monocentric chromosomes have a single centromere, which is the most common structure in most organisms. During cell division, the centromere serves as the attachment point for spindle fibers, ensuring accurate segregation. Dicentric chromosomes have two centromeres and can sometimes lead to chromosome instability. If both centromeres attach to spindle fibers, it can cause chromosome breakage during division, potentially leading to genetic disorders. Eccentric chromosomes lack a centromere, meaning they cannot attach to spindle fibers properly. As a result, they are often lost during cell division, making them unstable and typically non-functional. Polycentric chromosomes contain multiple centromeres spread along their length. Found in some plants and insects, polycentric chromosomes ensure proper segregation by distributing tension across multiple attachment sites. Chromosomes may also be classified based on the position of the centromere. In metacentric chromosomes, the centromere is centrally located, making the two chromosome arms almost equal in length. These chromosomes appear V-shaped during cell division. In submetacentric chromosomes, the centromere is slightly off-center, resulting in one short arm, called the P-arm, and one long arm, called the Q-arm. These chromosomes have an L-shaped appearance during division. In acrocentric chromosomes, the centromere is closer to one end, creating a very short P-arm and a much longer Q-arm. These chromosomes often have a satellite structure at the tips. In telocentric chromosomes, the centromere is at the very end of the chromosome, making it appear rod-like with only one visible arm. These are common in some species but not found in humans. The position of the centromere affects how chromosomes align and separate during cell division, ensuring the proper distribution of genetic material. Functions of chromosomes. Chromosomes contain the genetic material required for growth and development. It carries DNA, which contains genes that control all biological functions. These genes direct the production of proteins essential for the growth, development, and maintenance of an organism. From embryo to adulthood, chromosomes regulate cell function and development by carrying genetic instructions for cell division and trait maintenance. Chromosomes form the only link between two generations. It is the key to passing genetic information from parents to offspring. During reproduction, offspring inherit half of their chromosomes from the mother and half from the father, ensuring the transfer of traits. This process maintains continuity across generations and enables evolution over time. Chromosomes determine the sex of an offspring. In many species, including humans, specific chromosomes, called sex chromosomes, decide whether an offspring will be male or female. Chromosomes ensure the successful division of cells. For an organism to grow and repair itself, cells must divide properly. Chromosomes play a vital role in this process by ensuring that genetic material is accurately copied and evenly distributed during mitosis and meiosis. Errors in chromosome segregation can lead to genetic disorders, making their role in cell division critical for health and survival. If you enjoyed this video and found the information helpful, please like, subscribe, and share. Your support helps us create more content you love, makes it easier for others to discover our videos, and keeps you updated on our latest uploads. Thank you for watching. We truly appreciate your support.